So, good morning, everybody. Thursday again, and QBI. Today, we're going to talk about statistic, something about reproducibility, and how to handle your data. This is not in the title, but anyway, this will also come today. So first I need to load again some modules. That should be okay, I hope. Yeah, it's done. So again, we have some books that you can look at. The first two is are two old known ones. Then additionally, there is an overview chapter about statistics and hypothesis testing. And the last one is more about how to present your results graphically, visualize it in plots and different ways. There is also quite some papers to look at if you want to dig deeper down. And and then a few videos, some serious, some less serious, so a little bit entertaining is maybe this one, a guy who talks about how realistic some or useful some statistic studies are. Yeah, so this is the background material that you can dig deeper into. And until now, we actually quite far progressed in the class now, and until now we have talked about how to work with the images in terms of enhancement, understanding what the histogram is and how it works, automated methods for segmentation, basic segmentation first, and then automatic methods. Also, how to label different objects and then also do analysis on these objects. If they are very simple, small blobs, or if it's more distributed network shapes. And that is where we now come to what to do with this data, because now you have, say, tables of information, but that's you cannot say that there is a conclusion just from looking at these tables. So this is what we are going to look at, and mainly the statistical methods is as a central thing here. How to make a hypothesis around what the data you have got, and later also looking into how to do parameter sweeps. So that is for the testing, and later also how to actually pack up the data in data frames and how to visualize this data. So first, starting up with a little bit of background again, quantitative big imaging. So big data has been talked about for quite some years now. And I heard some managers say that big is when the data doesn't fit in a server rack. Oops, sorry. In a server rack or even in a server room, then it's big. But what does it mean for imaging? So first of all, it's not just large. So one tomography image is maybe 8 to 30 gigabytes. And then is the question, when do you think it's big? When it fits into your computer, when it fits into your workstation, or if you need a cluster for handling it. But important things for big data is the 3 V's: volume, velocity, and variety. And your methods need to be provided in a way that they can handle this. So they need to be scalable. They need to be fast, because if you have, I don't know how many terabytes or petabytes of data, then you need really to have algorithms and hardware to handle it in a decent way. Otherwise, you can probably wait to the next century until you have your data processed. And another thing is that your processing should be easy to customize so that you don't write a very narrow implementation that only works for your specific task right now, but maybe think a little bit wider and do it in a way that you can actually use it later. Now, looking at big imaging, what that is. So one thing is when we work with data, it's very important to work in a disciplined manner. So first thing is you need to have a recipe with well-described steps, what to do. And it should also be easy to regenerate the results. So for example, if first step is your professor says he doesn't really understand what that is really happening, are you sure your code is doing the right thing? Then it should be quite easy for you to just change in one corner and then run everything without having to click around for a week. Next thing is no magic. Hand waving and magic in reliable analysis is not really good. So you should try to avoid too many clickings and selections manually. The most things should be automatic and reproducible. And finally, of course, to understand what you have done, you need to document what your procedure. And the first step is what you write today, you don't remember in 3 months. That's kind of a fact. You need to really read through the code to understand. You may understand what you have done, but if someone is going to take over your code and work with your process, it's very hard to understand it from the code, and it takes very long time. So it's good to have some documentation telling what you have done. The least is at least is to document at least what the functions are doing that you have been working with, if you have written your own functions. So the advantages of working in this disciplined workflow is everything can be automated, hopefully. So it's for you, the overhead of doing 100 samples should be not much more than just doing a single one. So of course, you will have some initial effort to make it this way, but in the end, it should pay off. And actually, it mostly does. But it's easy to stray off and say, I need to improve it a little bit here, improve it a little bit there, and come into an evil loop of improvements. And that's not really what you want. So what you need to have is something that a really well-working script, and then more or less just modify few parameters, maybe the path to where the data is, and oh no, the screen is messing up again. Let's see, I think I steal this one. No. So let's try this one, if that works. Does it? No. Oh, there it is. Okay. So the parameters that you can work with is maybe one thing is where you have the data, but also maybe which type of cells you target or which specific characteristic you are looking for. But these should be really, say, abstract parameters that you easily can control. Then when you are setting up your study, you should be able to get some kind of numbers in the end. That's the goal of the whole experiment, not getting just beautiful pictures. And then you may need to make sure that your number is really meaningful. So that is actually what we are going to talk about today. And the question is, how can we compare the number different between different samples or different study groups of samples. Then is the question also within sample of the same type or between different samples. So there are different things you can compare. Then we also need to know a little bit how to compare different processing steps and like what is the effect of choosing this and that filter, this or that thresholding technique, what are the minimum volumes you get out, and which resolution is actually required to make a safe conclusion. So then we come into how to evaluate our parameter selection. And how can we be sure also that our technique is doing what it's supposed to do? But we have actually already talked about that in a couple of times actually in the filtering lecture where we did the smoke test, also in when we did the machine learning, where we actually did some tests against test data and validation data to be sure that we are doing the right thing. And finally, at some point, you usually want to pull up some kind of graph, if it's a bar chart or if it's a plot somehow, and how can we visualize all this data? In the beginning, we had this, I don't know, terabytes of data. And in the end, you want to visualize it in an understandable way to your audience. And showing a hard drive with terabytes of data is not really understandable. So we want to bring it down to some understandable way and be able to make conclusion from that. So how do we start? First, we look at our cell image and think about how we work with that. The first thing we did was to reduce the noise to make sure that we can find the cells we want to. That was in lecture 2 till 4. Then we analyzed the shapes and made sure that we actually looked at the right things. That was lecture 5 and 6. And then we also looked at some characteristics to look at the distribution of the objects. Now we have some samples, at least a collection, so we can get some statistics out of it. We get a large number of metrics. The question is which metrics actually make sense for your investigation. And then we have also, on the other hand, a lot of parameters that we need to tune, like filter parameters, thresholding, and so on. So let's go on with that. In your experiment, you have actually 2 choices: to look for correlation or for causation. Correlation is a quite easy thing. You see a very nice statistical relationship between some parameter in your process or in your sample and what the image features you want to observe. Causation is something that is a little bit harder because it's usually not easy to control. So that is when something happens in your in your object, and that results in a second happening. So that would be mechanism. There is some kind of mechanism that connects A and B, and it's very hard to show it, at least just from observations. And proving it out of observations only is very hard. So what we do, we have 2 types of experiments. Experiments where we just observe, and experiments where we control the procedure. And when we do an observation experiment, we have a random population. And it's not easy to do a hypothesis based on the observation experiment. And it's actually very rarely leads to some kind of causation. And then you can do some observations, like we examined 100 people, and we saw that there is a connection between blue eyes and that the average length is 10 centimeters, that these people are 10 centimeters taller. Another one would be we make 100 cakes, and we found a correlation between the baking time and the bubble size. That could be some observations that we can do. If we do a controlled experiment, which we actually mostly do, then we have some kind of specific groups that we work with, and these are controlled in some manner. It could be in biology that you check control different genes and see how they control have an effect on the growth, for example. And here you more easily can see the causation between that. If you change something, then you get this effect. So for example, we have 50 mice with some gene active and 50 other mice with the gene inactive. And we can see that the feet grows by 10%. Another thing is if we increase the temperature, the number of pores in some cast metal increase by 10%. That could also be examples of what you can get out of those experiments. So let's go to the statistics now. And statistics require a lot of data. It's usually in real examples, usually quite messy data to get some information out, and it's very specific and noisy. And that's not so easy to really teach and show what's going on. So we now go for a very simple model. We have a coin, the classic one, probably from any probability class that we flip. And we want to know if this coin is fair or magic or whatever. So first of all, we do some kind of experiments. If, for example, if I flip a coin 10 times and another one flips also the coin 10 times, would that be the same as 20 flips? So that could be a question that we could look for. Or else another one, if I flip the coin 10 times and then multiply it by 10, do I really have the same effect as from 100 flip? Rather not, actually. Or the other thing is you buy 10 of these coins and you want to know which ones are truly fair. And how can you handle that? Going into the little bit of the math of this task is we have each coin, which represents a stochastic variable. And each flip of that coin represents an observation of it. And then we have this x and xi, and the observation is the xi. And the act of performing the flip is f. So then we have the relation between the stochastic variable and its flipping, and its observation is this flipping here. In a fair coin, we would expect the probability for head or tail to be 50%. And that is what we hope for to get when we run it, this experiment. If not, then we can say that it is biased or magic or something.
Sorry, can I interrupt
the probability, yes?
I think we're stuck on the slides. I'm not sure. We're seeing slide 10.1. Is that the one we're supposed to be?
I'm on 19.1, actually.
Okay, well, then that's not the one.
No. Okay, I stop it.
We see your cursor, funnily enough, moving around, but on the wrong.
Okay, on the wrong place
slide.
In that case.
Yes.
So you see my screen now?
We see the screen, but still the same slide.
Oh, okay. Something stopped in the way. Okay, so do you see now the web browser?
Yes.
Okay, good. And now too?
Yes.
And now you see a slide with magic weighted coin?
I think it stopped while you switched. It's kind of stuck in between switching.
Ah, okay. Yeah, but do you see now experiment magic weighted coin?
Yes, but still on the browser window, kind of.
Oh, okay. So then maybe we can do it like this instead.
Yeah, now I see the slide.
Yeah. So this is okay. That's a bit unfortunate, but okay. Because now I don't have space on the whole. Yeah, then I have to make it smaller. So now you should see everything, I believe.
Yeah, I think it's kind of big for us, but we don't see the full slide, but most of it.
Not the full slide still?
Yeah, like 90% of the slide, I would say.
Okay, so you don't see the footer?
Yeah, correct.
Okay.
I don't see the last formula, but
the equation, do you see? The last equation with the product?
Yes.
Okay, then that's fine.
Okay. Good.
I'm not showing more.
Good.
Okay. So where was I? Yeah. Now on point 2, we are assuming that we have an unbiased flipper and that each flip is independent of the other. There shouldn't be any connection between them. And then we can say that the probability of the different flips together is just the product of them, or else we can also turn it into that it's the probability of each flip individually multiplied by each other. And if you have a good coin, then you should actually expect that it's the expected value of the flips should be the long-term expectation of the whole coin. So that is the expectation of the whole example here. So if we have this simple model and we want to compare it to reality, so first we have the coin flip, where you have each flip that provides you information about the coin, but also about the flipper. So the more flips we get some random stochastic variations in the coin, and also the flipper, all those variations, they cancel out. But if you have some systematic variation, they will accumulate and still remain in the measurement. If you would turn this into a real experiment, then we have that the experiment measurement tells us about, of course, our sample, which was the goal, but it also tells us about the instrument we used, if there is some variations in that, and also our analysis procedure. So more measurements now would provide, again, random stochastic variations in sample instrument analysis. They should cancel out. And actually, normally, the analysis shouldn't have too much stochastic variation. The few things in stochastic variations in the analysis is very minimal. So for example, rounding errors give you some kind of random effects, but not very much. And it's usually, compared to the other sources, it's not very much. And then, of course, you have the systematic variations in your instrumentation and analysis. They can actually contribute a lot. So looking at a little bit more complicated data, this is an old data set where we look at irises, different flowers. And here we have very many measurements from different plants and also some different species. So it's a pretty wide matrix in this data set.
Sorry, I think we're stuck again. It's still on the same slide.
No, come on. That's not fun. Okay. Also, when I do like this,
I don't see anything changing.
That's Zoom that doesn't like things. Okay.
Maybe I have to restart sharing on Zoom or something.
Probably. So let's try it again. Where is it? Here.
Yeah, now I see flowers.
Good. Beautiful, aren't they?
Yes, I hope it works now. Sorry for the interruption.
No problem. It should work. So you see the flip now?
Yes.
Good, good. So this data was collected back in 1936 and has been used in many statistical examples. And it's available. Here I use, again, a data frame, which is something I'm going to talk about after the break. And a little example out of this data set is here I picked just randomized, and you can see that they have different measurements on these flowers. And we also have different species. And each line corresponds to a different plant. And looking at the data to begin with, we use this pair plot where we look at combinations, pairwise combinations of the different measurements we have done. And in the diagonal, we have only the histogram of each variable. And you can see that in many cases, there is a separation, in particular between the red and the other ones. So there are red, blue, and some purple, which may not be so clear on the screen here in the room. But anyway, there's a clear separation between them in some variables and some variables less. So now we want to look at how similar different of these groups are. And we want to compare how similar they are within their own group and also how similar they are between the groups. And for that, we can use something called group similarity. And when we have low group similarity, the histograms are very much overlapping. It's harder to separate. And when we have high group similarity, the histograms are more separated and of course also then easier to separate. And here is an example with a sepal width. And sepals, they are these green leaves covering the flower bud. And they look like they are pretty much the same size in all these species. And of course, then they are overlapping also in the histograms. And that would be an example of low group similarity. If we look at petals, that's the beautiful leaves in the flower. They are very different between the different species. And you can see that each are nicely grouped and well separated. So here we have high group similarity. And this can be also quantified. So now I just used qualitative words. But it can actually be quantified using intra-class correlation coefficient. And that is the ratio between the standard deviation of the averages of each group divided by the sum of the standard deviation of the averages plus the average of the standard deviations. And with that metric, we can say that
sorry, I think you're muted.
you should hear me now. Good. I don't know what happened, but my microphone switched off. So first, when we start comparing groups, we have to make a list of assumptions. And out of these assumptions, we have to define a null hypothesis, which is usually denoted by H0. And that hypothesis is very often that both groups are the same. Then we calculate the probability of different observations given the truth of the null hypothesis. And that requires some knowledge about the distribution of the data. And also, that is actually a problematic thing because modeling of this can be very complicated. But very often you assume with many measurements that you actually have some Gaussian distribution due to the rule of the large number. And the outcomes of a decision making are different depending on what you want to decide. But usually you can base it on this matrix where retaining the null is the correct answer. Then you have the probability alpha, and then you have, in this case, you say that the population in the population is true, and rejecting the null give you a type 1 error. So that would be with some kind of probability alpha. In the other case, you can flip it, and then you have type 2 errors that you get the null hypothesis, but exactly you want to have the false. So then you have the other way around. And with probabilities, you also talk about the alpha is the type 1 errors or significance level. And that is a number that we are talking about very much soon. No. So going back to the loaded coin example, we have some assumptions. We flip and observe the coin accurately and independently, and all the coin is invariant and should have the same expected value. So that is our null hypothesis, is that the coin is unbiased. So the average of the flips should be a half. Now we can calculate the likelihood of such an observation given the number of flips. And with that, we get the p-value, which is what we are going to compare against. And the p-value is the likelihood that the null hypothesis is actually fulfilled. And you can see here with the Gaussian distribution that the p-value actually tells about this level here in the probability. And everything that's beyond it is very unlikely, and that's why we reject it. And for doing this, I said before that we assume that we have a Gaussian distribution, but it's with few samples, that assumption doesn't really hold. And the student's t-distribution is a way to actually model the uncertainty in our distribution. And there you have one of the parameters of this distribution is actually how many samples you actually use. And with that, we can make our hypothesis testing using the student t-test. And with that, we assume that we have an unbiased observations, and we also assume that we have normal distribution. And if the value produced by the student t-test is below some level, then we say, okay, this is not true anymore. And here I see I actually missed one thing that I thought I had, but anyway, that was the Python command to do this test. I think it's then in the notes. So back to the magic coin. So we say that if the p-value is less than 0.05, so 5%, then we can assume that this is a good base for rejection of the null hypothesis, where the null hypothesis is that the coin is fair. So if we get 5 heads, we should be good. 5 heads in a row. Then we should be able to say that this is not true. And looking at the test, that is very small, but anyway, looking at the test, if we increase the number of tosses, at some point we can say that the probability is really beyond realistic. And if we just flip one coin, then the probability is 50%. If we use the same equation, we come to 6.2% if we do n flips, 4 flips, sorry. And if we add one more flip and still get ahead, then we can start getting suspicious, because then we are below the 5% of the p-value. And how to calculate this when we have many experiments, then you can combine it. If you let more people toss the coin and so on, then we can combine it, that we have the probability of getting 5 heads when we do 5 tosses and we have 5 friends. And the first case, you can see that first case, 5 tosses, 1 person, 3.1%. But now something strange happens. We have 10 friends tossing the coin. All of a sudden, we get 27%. So then we couldn't reject this coin. And this says something is not right. And the reason is how we actually did the calculation compared to what we wanted to know. And what we wanted to know is that we didn't say that we want to get 5 heads ever. So that would be all the people that toss the coin, they get 5 heads. That's not really what we wanted. We want only that you have 5 tosses and you get 5 coins. And 5 heads, sorry. That would be the question we ask for. So we need to correct the whole thing by correction factor. And one thing we can do is to actually scale by the number of flips we do. And here we come into another example. Yeah, in principle, this is just about unrealistic statistical conclusions based on too much. Let's skip on to the next one. If we have now very many observations of very many variables, in this table, it's just randomized values, but we have assigned group 1 and group 2 to them. And now we want to investigate if the first section is different from the other one. And if you run the student t-tests on this, you can see that, yeah, here it is. That one was one I was missing. We can use in Python, we can actually do the t-test with 2 variables. We have variable in group 1 and in group 2. And when we do this test, we get different t-values, and we can see only one of these actually fall out as not fulfilling the null hypothesis. If we do it with even more, you can see that there are, in this case, it's not so easy to see right now. Yeah, when I run this cell, I would actually get two yellow lines here. It would be the first 2 here. They can be rejected, and the rest would be actually fulfilling and saying that these 2 variables are the same, which actually should, because it's actually the same random sampler we use. And an interesting thing is when you run many repeated experiment, you can see that there is some kind of trend that you have the number of significant variables found for one example, and repeating it 11 times, 21 times, up to 141 times. You can see that there is actually some kind of trend in the data. And that kind of trend is something we need to correct for when we do multiple tests. And here is a different way to visualize it. It's box plot, where you can see the different quantiles and some outliers. The diamonds here are the outliers. And now when we run many tests, we can see that the number of significant values actually increased with the number of variables that we study. And that's not right. So we need to correct our threshold level by the number of tests. And that is using a correction method introduced by Bonferroni. That test is criticized because it's too strict. Some people say that you need to do other tests, and there is actually a number of different ways to do these corrections. And but this is the easiest one actually to work with to give you a first idea. And if you will look at the results, we have two random variables. We have, in this case, done a study where we have two variables. One is at 0 average and one at average, one standard deviation away from the first one. And you can see in this example, we have the histograms of our two data, and that there is actually a difference in the average. And when you run this on very many variables, you can see in the beginning, the rejection is quite clear. But at some point, when you increase the number of variables, you can see that there's a higher likelihood that you actually would accept the null hypothesis the more variables you do. So that is the reason also why you need to do this scaling. This peak here is just the collected tail counts. So actually they would go much further away than I have visualized here. And yeah, you can see here also that it's actually this peak is increasing. So the message here is, in principle, if you do multiple variables that you want to investigate, you need to scale your confidence level in order to get the right results. And here you can see also the likelihood to find a different variable is actually decreasing with the number of tests. And with that, I think, yes, that would be time for a break. And I think the bell will probably ring in a few seconds.